Hi guys, welcome to my sister's house. Today we are going to be talking about chapter 9, which is all about microbial genetics. Okay, so this is going to be a ton of fun. So basically, we're going to start off with what are plasmids? Well, this is like the beginning of your chapter 9 book. This is that plasmids are a circular snippet of DNA carrying genetic treasures. And down here, you can see that this is a mating dance between cells that are going to be connected through these conjugation pili. So remember, pili are one of the appendages, right? Conjugation is when you conjoin or mate, right? And then down here, you can see the Actinobacter, which is a tiny non-motile gram-negative bacilli that comes to a hospital in your neighborhood. That would not be so much fun. So basically, what are we talking about in this chapter? Well, a whole bunch of genetics. So basically, chromosomes and genes, the packages of DNA, and all cells that are tightly, what are chromosomes? They're all cells that have tightly wound, they're elongated strands of DNA into compact chromosomes. And these would be genes that are bearing specific codes, and they're located at the sites along the length of the chromosome. So you can see here, and this is in the scoping out chapter, and like the first page of your book, that you have the DNA here, which is the deoxyribonucleic acid. That's what DNA stands for, is deoxyribonucleic acid. Well, here you can see how DNA is replicated, or we make copies of to replicate, right? Make a copy of that DNA. So basically, the codes that are carried by the DNA are conserved for our offspring and for future generations. And this ensures that the organisms will inherit the complete and accurate collections of genes that are required to survive and to reproduce. We then have transcription and translation, and this is how we interpret that blueprint. This is where the DNA acts as a model for assembling RNA copies of its codes. And the RNA are going to be transported to ribosomes to then read the coded message and express it into useful cell proteins. And so basically the shorthand formula for this is DNA turns into RNA, which then turns into protein, or codes for protein is probably a more accurate description. And then you can talk about mutations, and this is what happens when we have changes in the DNA code. Like any blueprint or language, mistakes can change the meaning of the DNA message so that transcription and translation, they're not able to produce the intended protein. And so with a variety of outcomes, these mutations can usually be pretty harmful. So then we can follow up and close this chapter with recombination. And this is a transmission of DNA from one cell to another. And some bacteria readily are going to share their DNA codes with other bacteria, and even with viruses, as seen in this very small picture. This results in bacteria with new genes and traits that help them adapt to their changes in environment. So basically, what is genetics? Genetics is the study of heredity, right? And so this is the science of genetics, and this is going to explore the transmission of biological traits from parents to offspring, the expression and variation of these traits, as well as the structure and function of the genetic material, and how this material changes. So when we're looking at the level of gene structure and function in the genome, this is figure 9.1, and this is called the levels of genetic study. So genetic processes can be observed at the levels of the organism, which would be here, the organismal level, the cell level, the chromosomal level, or at the molecular level, which would be the DNA sequence level. And though all the levels shown here are for a eukaryotic organism, they also follow a very similar pattern in the prokaryotic cells. So talking about some key words with microbial genomics. A genome, well, what is that? That is the sum of total genetic material, or DNA, in a cell. And most exist as chromosomes, and some appear in non-chromosomal sites, such as mitochondria, chloroplasts, or plasmids. And the genome of the cell has DNA, where the genome of viruses can have either DNA or RNA. In the very beginning, when we talked about viruses, I told you that they were kind of tricky, and this is one of the reasons why. Okay? Oh, my computer just decided to freeze. All right, there we go. So what is a chromosome? Well, this is a discrete cellular structure that is composed of neatly packaged DNA molecules. And the eukaryotic chromosomes are located in the nucleus, and they are multiple, meaning there's more than one, and they are linear or in a line, where bacterial chromosomes are a singular loop. So looking at this picture, this is picture 9.2 in your textbook, and this is the general location and forms of the genome in 2 cell types and selected viruses. And of course, this is not drawn to scale. And so this would be your eukaryotic cell here, where your prokaryotic, remember, these are kind of like your primitive cells. So they're kind of just floating out there in the cytoplasm, where in the more organized eukaryotic cells, they're going to be contained within the nucleus. So basically, genotypes and phenotypes, okay? Genes are going to be the genetics. Phenotype, I like to see the word photo in that word. So a phenotype is going to be how we look, okay? So my genotype is what's going to code for like my hair color, and my phenotype would be that it's brown, okay? So a chromosome is going to be divided into genes, and the gene is the fundamental unit of hereditary, which is responsible for a given trait. So the site on a chromosome that provides the information for a certain cellular function is called a gene, and the segment of DNA that contains the necessary code to make a protein or RNA molecule is also that gene. And there are 3 basic categories of genes. Genes that encode for proteins, and these would be your structural genes, genes that encode for RNA, as well as genes that control gene expression. And those kind of regulate the body, so those would be your regulatory genes. Okay? So all types of genes constitute the genetic makeup, and this is called the genome, okay? And the expression of the genome is going to create observable traits. And like I said before, it's kind of like a picture. That is the phenotype. Okay? So when we talk about the size and the packaging of the genome, the smallest virus has about 4 or 5 genes, where E. coli, which we have studied already in the lab, this is a single chromosome that contains 4,288 genes. It's only 1 millimeter big, and it's 1,000 times longer than the actual cell. It's just coiled up on itself. Where in the human cells, we have 46 chromosomes that contain 31,000 genes, and each of them are 6 feet, and this is 180,000 times longer than the cell. So when they're really packed together, they can coil up really easily. So then you can see here in this picture down here, this is a disrupted E. coli cell. And this cell has been ejected and it has unejected and unraveled chromosome, which now appears as thin strings radiating around the outer surface. So all these little wispy guys, that would be the expelled genetics. So DNA molecule is compacted in the cell by supercoil or super helices. And you can think of a helix as like a staircase, right? It's 2 things that are wound together in like kind of a spiral fashion. So in prokaryotes, by the action of the enzyme DNA gyrase, this coils the chromosome into a tight bundle by a reversible series of twists in the DNA molecule. Where in eukaryotes, the packaging is more complex because they're more complicated cells. And with 3 or more levels of coiling, we have starting with a chain of the nucleosomes, and these DNA particles, strands, are wrapped around histone proteins. So you can see here that this is the packaging of DNA, and this is figure 9.4 in your book. So eukaryotic DNA is going to interact with a variety of different proteins and small molecules. This relationship regulates the expression of many genes while at the same time allowing for the ordered condensation of DNA into a compact supermolecule. Okay, so a condensing up. So you can see that the condensed metaphase chromosome looks like an X, but if you look closer, it's coiled. You look closer, it's loosely coiled. You look closer, it's a chromatin fiber, so it's even tightly spiraled. And then you look even closer, and they're going to be strands of DNA that are wrapped around these histones. And basically, the histones just kind of help things keep untangled, right? So they help to keep it nice and orderly. So what is the structure of DNA? Well, this is that double helix. And so the basic unit of DNA is going to be the nucleotide. Okay, let me move out of the way. And this is a deoxyribose sugar and a phosphate group. And that's going to be your backbone. And then you have your nitrogenous bases, and those would be adenine, guanine, thymine, or cytosine. And these nucleotides covalently bond to form a sugar phosphate backbone. And so this would be a picture of your double helix structure, and this is figure 9.5. So this is a simplified structure of DNA where nucleotides stack together to form two opposing strands that are held together by hydrogen bonds here in the middle between the base pairs. Well, nitrogenous bases covalently bond to the number 1 prime carbon on each sugar, and then they span the center of the molecule to pair with a complementary base on the other strand. So you have to remember that A will always bind with T. C always binds with G. So A, T, C, G. Some of my other students say, well, apples grow on trees and the cars go into the garage. So apple, tree, car, garage, A, T, C, G. And the reason is because adenine and thymine have 2 hydrogen bases, where guanine and cytosine have 3 hydrogen bases. So the shape just works better for them, okay? And so this would be your picture there of the double helix, and this is going to be figure 9.6. And so this specifically is a space-filling model of DNA that shows the basic configuration of a double helix of a sugar phosphate backbone that holds the nitrogenous bases between them. Okay. So DNA double helix also has anti-parallel arrangement. So that means if you think of parallel parking, I have a big old truck. If I'm trying to parallel park, I'm facing in the same direction as the road, right? Or the sidewalk, where anti means the opposite. So anti-parallel means you're going the opposite way, kind of like driving down the road, right? We're going on one way on the road, and the other side of the road has cars going the other way. And so this is like on the top strand, we're going to go from 5' to 3'. And since it's complementary and anti-parallel, that means that the bottom strand then is going to look like it's going from 3 to 5. So each strand provides a template for the exact copying of a new strand. And the order of bases constitutes the DNA code. And so this would be, again, still in figure 9.6. And this is the twisted ladder model. And this depicts how the complementary nitrogen bases, which are the bars, the blue bars on the side, pair with each other across the center of the molecule. Okay. So what is the significance of this? Well, it's to maintain the code during reproduction. So consistency of base pairing guarantees that the code will be retained. When the strands are separated, each strand is going to serve as a template for replication of the molecule into an exact copy. And this also can help provide variety. So the order of bases responsible for RNA and protein synthesis, therefore for the phenotype of each organism, is encoded by this. And so like I'm at my sister Corinne's house, she has blonde hair and blue eyes. She has straight hair, I have curly hairs. So that means, I mean, we have the same genetic basis, we have the same parents, but there's variety with how our genes were reproduced, and that makes us us, okay? So for your first concept check, in DNA, adenine is a complementary base for what, and cytosine is what? So remember that the apples go in the trees and the cars go in the garage. So adenine and thymine, cytosine and guanine would be your correct pair. So what is the overall replication process? Well, replication occurs on both strands simultaneously, so at the same time. And this is a semi-conservative process. This means that the parent DNA molecule is uncoiled or unraveled, because remember, they're tightly spiraled around those nucleosomes. Then those two strands, because it's a double helix, right, are going to be separated, and that's going to expose the nucleotide sequence, ATCG, right, to serve as the template. And then we have two new complementary strands that are going to be synthesized by using each single-stranded template as a pattern. And that can be seen here. And this is figure 9.7. So this is simplified to show the semi-conservative replication of DNA. So the two strands of the parental DNA double helix are unwound and separated. And each single strand will serve as a template, which you see in blue here, to synthesize or make a new strand of DNA, which is shown in red. Or pink, reddish pink, right? And so starting at the replication fork, And as synthesis proceeds, the correct nucleotides are added in according to the pattern of the template. An A on the template will pair on T on the new molecule, otherwise called a daughter strand, and C will pair with G. The finished daughter strand of DNA molecules then contain one strand of the newly made or newly synthesized DNA and one of the original parental strand. And this keeps the code intact because the same linear arrangement of the bases is maintained during the process. And this process is covered in greater detail coming up in figure 9.8. So all chromosomes have a specific origin of replication site as the place where replication originates or is initiated. The origin of replication is AT or adenine thymine rich. So therefore, less energy is required to separate these two strands. And there are two replication forks where DNA is being synthesized. Each contains its own set of replication enzymes. So here would be your picture of the replication fork. And this is figure 9.8. So the assembly line of DNA replication in a circle bacterial chromosome, which is in your book shown down here. So a bacterial chromosome showing the overall replication. There are two replication forks where DNA is being synthesized, right? And then here, this is B. This is an enlarged view of the left replication fork to show the details of replication, okay? So you can see at 1 that the chromosome to be replicated is unwound by a helicase, which forms a replication fork with two template strands. And then in step 2, which is over here, the template for the leading strand, which is blue, is oriented 3 to 5. So this allows the DNA polymerase to add nucleotides to the 5 to 3 direction in the replication fork so that it can be synthesized or made as a continuous strand. So note that the direction of the synthesis refers to the direction of the new red strand. Then we go up to step 3, which you can see here. And so this is the template for the lagging strand, and this runs 5 to 3, which is in the opposite direction of the leading strand. So to make a new strand in the 5 to 3 orientation, synthesis must be proceeded backwards, away from the replication fork. So then in 4 here, before synthesis of the lagging strand can start, a primase is going to add an RNA primer to direct the RNA polymerase. And synthesis produces unlinked segment of RNA primers and new DNA that's called Okazaki fragments. Okay, so basically, since we're going backwards, we can't walk very efficient backwards, so we take it one step at a time. Same thing here. We're making little chunks of DNA and then putting them together. And those are called Okazaki fragments. And that's a very fun word to say. It's one of my favorite science words. Okay, then here in 5, you can see that DNA polymerase 1 removes the RNA primers and it fills in the correct complementary DNA nucleotides at the open site. And then over here in 6, then the unjoined ends of the nucleotide, which is a nick, must be connected by a ligase. And to ligate is a fancy word of to glue. Okay? So the figure of DNA replication, like I just read all of this to you, okay? So I read it to you when we were going over that picture, but here are the steps if you want to read them, okay? So step 1, 2, 3, 4, 5, and 6, okay? So again, remember my favorite word is Okazaki fragments, okay? No, that's not going to necessarily be on the test that it's Janet's favorite word, but it is a very cool word. Okay, so then making an exact duplicate of DNA involves 30 different enzymes. So it's quite a process, okay? Helicase is going to unwind and unzip the DNA double helix. I probably shouldn't tell this joke, but it's really funny. Scientists are kind of nerds, and so we say, I wish I was DNA helicase so I could unzip your genes. Ha ha ha, Genes, like genetic genes. Okay, but that's just kind of a fun way to remember that one. So the helicase is going to unwind and unzip the DNA double helix. And then the RNA primer is going to be synthesized or made at the origin of replication by a primase. And then DNA polymerase 3 then adds nucleotides from a 5' to 3' direction. And this is the leading strand, and this is going to be made or synthesized continuously in the 5' to the 3' direction, where we have that lagging strand, which again is in 5 to 3 in short segments because it's like walking backwards. So the overall direction is in 3 to 5 because we're putting together segments together in the 3 to 5 direction. Then DNA polymerase 1 removes the RNA primers, and it's going to replace them with DNA. So this is the other part of that figure of 9.81. And so this is that part A that I talked about, even though it's labeled B. Okay, well, B is the enlarged view of the left replication fork to show the details of replication. Okay, so this is what's going to happen. We have the separation that's occurring and then the replication that's going to occur. Okay, so when replication forks meet, ligases ligate or glue link those DNA fragments along the lagging strand. As replication proceeds, one newly made strand is going to loop down. And when the forks have gone a full circle, a termination site shuts the replication down. And those two circular daughter molecules remain connected briefly, but then they are nicked and separated by a helicase. And so this would be figure 9.9, and this would be the completion of a chromosome replication in bacteria. So in A, up at the top, this is when, as the replication proceeds, one strand loops down. Where B, the final separation is achieved when the enzyme cuts and releases two completed molecules. And each daughter cell is going to receive one completed chromosome during binary fission coming together. Okay. So here is a fun little summary for you, and that will help you to learn these things. And so these are some enzymes that are going to be involved in that DNA replication so that you can have it all, this is table 9.1, so that you can have them all in a nice little tidy bunch. So here's another concept check. Why must the lagging strand of DNA be replicated in short pieces? Well, remember, it's because the DNA polymerase only works in the 5' to 3' direction. So this is like walking backwards. We're still making forward progress. We're just doing it one step at a time backwards, okay? So genetic information in the DNA molecules is conveyed to RNA through the process of transcription. And the information that's contained in the RNA molecule is then used to produce proteins in a process that's called translation. So a wide variety of specialized RNA acts by regulating the gene function. And so here is an example of the summary of the flow of genetic information in cells. And this is figure 9.10 in your book. And DNA is the ultimate storehouse and distributor of genetic information. DNA must be deciphered into usable cell language. It does this by transcribing its code into the RNA helper molecules that translate the code into a protein. And then in B, the other sections of DNA are copied to make additional types of RNA molecules that then regulate the genes and their products. Okay? So what about gene-protein connections? Well, each structural gene is an ordered sequence of nucleotides that codes for a protein's primary structure. Their groups consist of 3 consecutive bases, and these are called triplets or codons. More commonly, they're called codons. On one DNA strand, they are transcribed into RNA sequence triplets. Each triplet of nucleotides on the RNA specifies a particular amino acid. And a protein's primary structure, which would be the chain of amino acids, is going to determine the shape and the function of that protein. Okay? And proteins contribute to the cell phenotype, or the way that it looks, as enzymes and structural proteins. So this is figure 9.11, and this is a simplified view of the DNA-protein relationship. So the DNA molecule is a continuous chain of base pairs, but the sequence must be interpreted in groups of 3 base pairs, which is called either a triplet or a codon. And each triplet is transcribed into mRNA codons, and this will translate into one amino acid. Consequently, the ratio of base pairs to amino acids is 3:1. It takes 3 letters to make one amino acid. So when we're talking about RNAs, these are major participants in transcription and translation. And the general structure of the ribonucleic acid, or RNA, is different than that of the DNA molecule in many different ways. One is that the RNA is going to be single-stranded molecule that can assume secondary and tertiary levels of complexity, leading to specialized forms of RNA, such as messenger RNA, tRNA, and rRNA. The RNA, I say it's a little bit of a diva, and it's like, I'm way too important for T's, so no, you must use U's. So RNA contains uracil, Therefore, it has no T's, like DNA. But RNA tells you that it's RNA, so that's not really that hard to remember because it's like mRNA, tRNA, rRNA. So it's like, ha 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 ha, I'm RNA, don't use T's, okay? So just remember that RNA has no T's, it just has U's. And then the sugar in RNA is ribose rather than deoxyribose, okay? So kind of think of D as di, 2, right? Where ribose is just 1. And so here is a nice little summary table, and this is table 9.2. And these are major types of ribonucleic acids that are involved in protein synthesis. Okay. So what about messenger RNA? It's one of those specialized RNAs. Well, this is a transcribed version of a structural gene or genes in DNA. And this is made or synthesized following a complementary base pairing by a process that's similar to the synthesis of the leading strand during DNA replication. And the message in triplets is called codons. Okay? And so this is going to be figure 9.12, and it specifically shows you messenger RNA. And so mRNA is a short piece of messenger RNA, and this is going to illustrate the general structure of RNA. It is a single strand repeating phosphate ribose sugar backbone, which is going to be attached to a single nitrogenous base. And again, it's going to be, instead of AT, it's going to be AUCG. Okay, and so that would be your messenger RNA. Well, then we have tRNA or transfer RNA, and this acts as a translator of the mRNA code into a protein. And these are about 75 to 95 nucleotides in length that are bent into a hairpin loop that form like a cloverleaf structure that is further packed into a complex helix, right, or a spiral. The bottom loop of the cloverleaf is going to expose the tRNA-specific anticodon, which is complementary to an mRNA codon. And the binding site for amino acids is specific for each anticodon. Well, then we have tRNA, because we were just talking about, well, the second part of tRNA. And here's that loop that it was talking about. So there's the picture that I'm going to describe here. And again, this is figure 9.12 in your book. So the tRNA on the left is going to be the strand loop that loops back on itself to form that interchain hydrogen bonds. The result is a cloverleaf structure that's shown in the simplified form. And at the bottom is that anticodon that specifies that the attachment of a particular amino acid at the 3' end. And then on the right, we'll see here in a second, is a three-dimensional view of the tRNA structure. So here on the left, it's showing you that cloverleaf, where on the right, it's showing you that hairpin. Okay. Well, then we have ribosomal RNA, or rRNA, and this is the prokaryotic 70S ribosome that we talked about when we talked about 70Ss before. This is a particle that's composed of a tightly packed ribosomal RNA, or rRNA, and a protein. And this forms a complex three-dimensional figure that contributes to the structure and function of ribosomes. And so what do ribosomes do? Well, they read the messenger RNA code, they facilitate or help the interaction with tRNA, and then they produce proteins at a very impressive rate, which means fast. Okay, so this little blue guy is figure 9.14. And then again, this is going to be the players in translation. So a ribosome serves as a stage for protein synthesis, and the assembly of the small and large subunits results in specific sites for placement of the mRNA, which would be the small subunit, and the 2 tRNAs with their amino acids, which would be in the P and the A sites. So backing up a little bit to talk about transcription. This is the first stage of gene expression. So during transcription, an RNA molecule is going to be made using the code on DNA as a guide or a template. And it's going to proceed in 3 different phases. You have initiation, which is to start. And so this is when the RNA polymerase is going to bind to the promoter region upstream of the gene. Then you have elongate. If I elongate my neck, right, giraffe neck, well then it's making it longer. So this is when it's growing. So the RNA polymerase is going to add nucleotides that are complementary to the DNA template strand in the 5' to 3' direction. Remembering when we're dealing with RNA, uracil is going to be complementary to adenine. And then you have termination, kind of like the Terminator, right? So that's stopping. So this is when the RNA polymerase is going to recognize a stop sign in the DNA, and it's going to release the transcript. And this can be anywhere from 100 to 1200 bases long. So the figure 9.13 is the one that they're talking about, and the figure of major events of transcription. And so for number 1, it says each gene contains a specific promoter region and a leader sequence for guiding the beginning of translation. Next is the region of the gene that codes for the polypeptide, and it ends with a series of terminal sequences that stop translation. And then DNA is unwound at the promoter by the RNA polymerase. And only one strand of DNA, which is called the template strand, supplies the codes to be transcribed by an RNA polymerase. And then this strand runs in the 3' to 5' direction. Moving on to step 3 then, the RNA polymerase moves along the DNA strand, and this adds complementary nucleotides as dictated by the DNA template. And then the mRNA strands read from a 5' to 3' direction. And then step 4, which is down at the end, the polymerase continues transcribing until it reaches a termination or a stop site. And then the messenger RNA transcript is released to be translated. Note that the section of the transcribed DNA is rewound into its original configuration. So here is that picture, right, of step 1, 2, 3, and 4 that we just talked about. Okay. And again, this is figure 9.13. So then this is going back to that figure that we just talked about, 9.14 previously, and this is where we're talking about translation, and this is the second stage of gene expression. So all of the elements needed to be synthesized of the protein are brought together on the ribosomes, and then the process occurs in 5 stages. You have the initiation or the starting, the elongation or growing, the termination or stopping, and then you have the protein folding and processing so that things can function properly. And the master genetic code is represented by mRNA codons and their specific amino acids. And this code is universal among organisms, and it is redundant. So this table is the master genetic code, and this is figure 9.15. And this is basically like the mRNA table, okay? So the codons of mRNA that specify a given amino acid. So if I had, for instance, ACA, I would look for A over here in the first position. I would look for C here in the second position. I would see where those two intersect, look for the third letter. So ACA would be threonine, okay? So how do we interpret this genetic code? Well, this is in figure 9.16. And so if the DNA sequence is known, the mRNA codon can be surmised. If the codon is known, then the anticodon and finally the amino acid sequence can be determined. The reverse is not possible, so we can't determine the exact codon or anticodon from an amino acid sequence due to the redundancy of the code. Okay, what that means is like serine is coded by AGU and AGC. So we would just know that it was serine, but we wouldn't know if it was AGU or AGC, right? It just we know that it comes from serine if we're reading it backwards. So speaking specifically about translation, well, the ribosomes assemble on the 5' end of the mRNA transcript. And then the ribosome scans the messenger RNA until it reaches the start codon, and this start codon is usually AUG. Then a tRNA molecule with a complementary anticodon and methionine amino acid enters the P site of the ribosome, and it binds to messenger RNA. Well, then we have the entrance of tRNAs 1 and 2, and this is the process figure 9.17. And so step 1 is the entrance of tRNAs 1 and 2. And then the second tRNA with a complementary anticodon fills the A site, and then a peptide bond is going to be formed between the amino acids on the neighboring tRNA, and this forms that peptide bond. And then the first tRNA is released, and the ribosome slides down to the next codon. And then from there, another tRNA fills in the A site of a peptide bond is formed. And this is the first translocation. The tRNA2 shifts to the P site, then the tRNA3 enters at ribosomes A. So it's kind of like hungry, hungry hippo. It's like chunk, chunk, right? And it's just going to kind of chunk through it, okay? And then the fifth one is going to be the formation of a peptide bond. And you can see that we have our growing tail over here. Okay, or I'm sorry, actually up here. So this is where it's coming off of and it's forming these amino acids down here. Okay, so this process continues until a stop codon is reached, and then we discharge tRNA2, and then the second translocation, tRNA4, is going to enter that ribosome. And then we form that polypeptide bond here to link it together. So remember, chunk, 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 right? And yes, you have to make those noises when you do this, okay? And then we have those termination codons, right? And these would be UAA, UAG, or UGA. And these are the codons for which there's no corresponding tRNA. So when the last codon is reached, the ribosome falls off, and then the last tRNA is going to be removed from that polypeptide. So for a concept check here, if we have the tRNA, what is it? It is the molecule that what? Pause and come back. Well, tRNA acts as a translator of the mRNA code into a protein, okay? So then moving on to the Polyribosomal Complex. This is figure 9.18, and this is speeding up the protein assembly line in bacteria. So in A, which is going to be up here on the upper left, the mRNA transcript encounters the ribosomal parts immediately as it leaves the DNA. And then in B, down here, we're going to have the ribosomal factories assemble along the mRNA chain in a chain, and each ribosome reads the message and translates it into a protein. And many products will thus be well along in the synthetic pathway before transcription has even been terminated or stopped. And then in C, over here, you can see a photomicrograph of a polyribosomal complex in action. And note that the protein tails vary in length depending on the stage of translation. Okay, so the tails are different lengths. So eukaryotic transcription and translation do not occur at the same time. Transcription occurs in the nucleus and translation occurs in the cytoplasm. The eukaryotic start codon is AUG, but it does not use the formal methionine. Eukaryotic mRNA encodes a single protein, unlike bacterial mRNA, which encodes many. And then eukaryotic DNA contains introns, which intervenes the sequences of non-coding DNA, and these therefore have to be spliced out of the final mRNA transcript. We used to call this junk DNA, but we actually find that there's some genetic codes in it now. I digress. Okay, so then what are we doing when we're splicing that eukaryotic pre-mRNA? Well, eukaryotic gene coding sequences, or exons, are interrupted by segments that are called introns. So this one's kind of vice versa. We keep the exons and we kick out the introns. Okay, so just kind of remember it backwards. So introns are transcribed, but they're not translated, and therefore they're going to be removed before translation. And splicing does not occur in prokaryotic cells. So this is figures 9.19, and this would be the split genes of eukaryotes. So eukaryotic genes have an additional complication in their transcription and translation. Their coding sequences, or exons, which are located E, or they're demarcated E here, are interrupted at intervals by segments that are called introns, or the ones that are labeled I, and these are not part of that protein's code. And so the introns are transcribed, but they're not translated, which necessitates their removal by RNA splicing enzymes before that translation can occur. So then we regulate the protein synthesis and metabolism. So genes are regulated to be active only when they're needed. Okay, when those products are needed, then it's like soldiers, they're called battle. Okay, and then in prokaryotes, this region is coordinated by operons, or a set of genes, all of which are regulated as a single unit. So there are two types of operons. The first one is inducible, where the second one is repressible. Okay, so inducible operons, this is when the operons are turned on by the substrate. These are catabolic or breaking down operons, and those enzymes are needed to metabolize a nutrient, and they're produced when needed. We also have repressible, and these are genes that are in a series that are turned off by the protein that's being made. And these are anabolic or building operons. And these are enzymes that are used to synthesize or make an amino acid stop being produced, and they stop being produced when they're not needed anymore. So it's kind of like an on-demand system. So these are made up of three different segments. We have a regulator, and this is the gene that codes for the repressor. We have a control locus, and a locus is just a fancy word for location. So you have a control location, and this is composed of a promoter and an operator. And then you have your structural locus, and this is made of three genes each, and they code for an enzyme that are needed to catabolize lactose. So we have beta-galactosidase, and this is going to hydrolyze lactose. We have a permease, and this is where it's going to bring lactose across the cell membrane. Then you have beta-galactoside transacetylase, and this, we don't exactly know the function, but we know that it's there, so we have to learn it. Okay, so the Lac Operon is talked about a lot, and this one's usually off. And so in the absence of lactose, the repressor binds with the operator locus or site and blocks transcription of downstream structural genes. So in this one, when we have the operon off, then we have no lactose that's being made. Okay, so this is figure 9.20. Part 1. And so in the absence of lactose, a repressor protein, which is the product of a regulatory gene located elsewhere, attaches to the operator of the operon. And this effectively blocks transcription of the structural genes downstream to its right. And then there's suppression of transcription and translation, and this is going to prevent unnecessary synthesis of enzymes for processing lactose. The lactose turns the operon on by acting as the inducer. So the binding of lactose to the repressor protein changes its shape and it causes it to fall off the operator. Well, RNA polymerase can bind to the promoter and then structural genes are transcribed. And so with the figure, again, this is figure 9.20. When the operon is on, then lactose is present. And upon entering the cell, the substrate or that starting material, which is lactose, becomes a genetic inducer by attaching to the repressor, which is rendered inactive and therefore falls away because it's not needed. The operator is no longer closed off and its DNA becomes accessible to the RNA polymerase. Well, then the RNA polymerase is going to transcribe that structural gene, and the mRNA is translated into the enzymes that can therefore act on the lactose substrate. And so this would be a figure, again, of that Lac Operon turning on here. Okay. And so, so upon entering the cell, the substrate lactose becomes genetic inducer by attaching to the repressor, which is rendered inactive and falls away. Operator no longer closed off. Everything I just said. Okay. And then we have Arginine operon, and this is repressible. And so it is normally on, and it will be turned off when the product of the pathway is no longer required. And this figure of Arginine operon on. Okay, and so, yes, and so the operon on, this is when we have the Arginine, I'm trying to find which figure this is. They don't really have it. Okay, so the Arginine is being used by the cell. So a repressible operon is going to remain on when its nutrient products, for instance here, arginine, are in great demand by the cell. And so the repressor has the wrong shape to bind the DNA operator without a co-repressor, so that the RNA polymerase is free to actively transcribe the genes and translation actively proceeds. Okay, and so this is again going to be, this is figure 9.21, part 1. So this is when the operon is going to be on, and it continues on this slide. Okay, so the operon on, arginine being used by the cell, a repressible operon remains on when its nutrient products, which again here is arginine, are in great demand by the cell. The repressor has the wrong shape, etc., so it can't bind. Okay, computer, wake up! My computer decided to go to sleep. When excess arginine is present, it binds to the repressor and changes it. And then the repressor binds to the operator and blocks arginine synthesis. And then arginine is then going to be the corresponder. Okay, and so then we have operon off, and this would be part 2 of figure 9.21. And this is when the operon is repressed when arginine levels build up, and it serves as a co-repressor, and it activates the repressor. Then the activated repressor complex affixes to the operator and it blocks the RNA polymerase. Therefore, transcription of genes for the arginine synthesis is turned off. So again, this is part 2 that we just talked about. So here's a little concept check for you. Okay, and so if the operon's repressor is in active form, that means that the transcription from the operon is occurring. Okay, sorry, this is pick A or B. So if an operon's repressor is in its active form, that means what? Either A, the transcription from the operon is recurring, computer stop, okay, or transcription from the operon is not occurring. Let me try to, whoops, I didn't want to do that. All right, and so the correct answer would be, it would be B. So when it's in its active form, it means that transcription from the operon is not going to occur. Okay, so then talk about mutations. What happens when things go wrong? So changes in the genetic code. A change in the phenotype due to a change in the genotype, which would be the nitrogen bases sequence of DNA, is called a mutation. Okay, my computer is being silly. All right, and a natural non-mutated characteristic is known as a wild type or a wild strain, where an organism that has mutation is a mutant strain. And this shows variants or changes, different varieties of morphology, which is shape, nutritional characteristics, and genetic control mechanisms, as well as resistance to chemicals, etc. So how do we isolate mutants? Well, replica plating techniques allow for identification of mutants because the culture is exposed to the mutagen. The isolated colonies are transferred on a master plate, and then a tiny clump of the cells are picked up and transferred to a plate with a complete medium and another with incomplete medium. And then the colonies in the complete medium plates missing from the incomplete ones are mutant colonies that can be subcultured for more use. Okay, so this would be your picture of isolating the mutant. And this is figure 9.22. Okay, so it shows that this technique was developed by Joshua Lederberg, and it's based on A, which is up at the top. Let me see here, over here, right there, that is A. And this is exposing a culture to a mutagen. And then you're going to be plating the mutagen, which is here, plating the culture for isolated colonies on a master plate with complete nutrients. And then for C, which is going to be here, you are going to press the sterile carrier onto the surface of the colonies to pick up a tiny clump of cells and press it again to inoculate a plate of complete medium and then a plate of incomplete medium. And then down here, and D, you have after growth, the two plates are going to be compared, and the colonies that occupy a specific position on the complete medium but are missing from the same position on the incomplete medium are mutant colonies, therefore, that can be subcultured down here for further use. Okay? So what causes these mutations? Well, we can have spontaneous mutations, and these are spontaneous, right? So these are random changes in the DNA due to errors in replication that occur without a known cause. We also have induced mutagens, and these result from exposure to known mutagens, which would be physical, like primarily radiation, or chemical agents that interact with the DNA in a disruptive manner. Okay, so here is a fun little table. This is table 9.3, which lists selected mutagenic agents and their effects. Okay, so ethidium bromide, bisulfate, radiation, ultraviolet lights, etc. Okay, so there are categories of mutations, and these would be point mutations that have an addition, deletion, or substitution of a few bases. You have missense mutations, which causes a change in a single amino acid. You have nonsense mutations that changes the normal codon to a stop codon. You have silent mutations, and these alter a base, but it does not change the amino acid. You have back mutations, and this is when a mutated gene reverses back to its original base composition. Then you also have frame shift mutations, and this is when the reading frame of the mRNA is altered by either adding or deleting a letter. Okay. So figure 9.4 is a nice little table that summarizes all this. Well, wild type is going to be that non-mutated sequence. An example would be if ATCG, we're using letters here so it can spell out. So the example is the big bad cat ate the fat red bud in the wild type sequence in the gene, the DNA sequence found in most organisms and is generally considered the normal sequence. Where if we have substitution, we have a missense, we change one letter, so it ends up causing a different amino acid to be incorporated into that protein. And so these effects range from unnoticeable to severe based on how the new amino acid alters that protein shape and therefore function. You have the nonsense mutation, and that means that you have a codon that's going to be converted to a stop codon. This is going to result in a premature termination or stopping of the protein synthesis, and the effects of this type of mutation are almost always very severe. You have inversion mutations, and you can see here it kind of like jumbles it all around. So an inversion arises when adjacent letters exchange places, and this is going to alter either one or two bases depending on the location and the inversion. This does not change the reading frame, but it can result in significant changes in the amino acid and protein function. Frameshift mutations are going to be just an insertion or a deletion of a single letter. So the insertion or deletion causes a change in the reading frame of the mRNA, and this results in a protein which every amino acid after the shift can be affected. Because of this, frameshift mutations almost always result in a non-functional protein because it's not going to make sense. Okay, it's gobbledygook. Well, we have repair mutations as well. So since mutations can potentially be fatal, our body tries to catch them. And the cell has a lot of different enzymatic repair mechanisms in place to find and repair these damaged DNA. We have DNA polymerase, and this is going to proofread nucleotides during DNA replication. You have mismatch repair, and these locate and repair the mismatched nitrogenous bases that were not repaired during the DNA polymerase. Then you have light repair, and this is specific for UV light damage. You also have excision repair, and this locates and repairs the incorrect sequences by removing the segment of the DNA and then adding the correct nucleotides. We also have this thing that's called the Ames Test. And this is where any chemical that's capable of mutating bacterial DNA can similarly mutate mammalian DNA. And so agricultural, industrial, and medicinal compounds are screened using this Ames test. And this is an indicator organism that is a mutant strain of the Salmonella bacterium that has lost the ability to synthesize or make histidine. So this mutation is highly susceptible to back mutation. Okay, so here is a picture of the Ames test, and this is figure 9.23 in your book. So you can follow along as I read the title. So in this traditional plate method, a strain of Salmonella enterica that cannot synthesize histidine is the test species. And then the chemical causes an increase in mutation rate to histidine-producing cells as compared to a control. And the chemical is considered mutagenic. Okay, and then B, the newer well fluctuation test is based on the same scheme. However, it has a greater sensitivity and it is easier to evaluate. So this is traditional. This is the newer well plate technique. So positive and negative effects of these mutations. Well, mutations can lead to non-functional proteins, and these can be harmful and potentially fatal. And organisms with mutations that are beneficial in their environment can rapidly adapt, survive, and reproduce. And these mutations are on the basis of the change in population. For instance, they think that giraffes originally had a shorter neck, but because of nutrient availability, the ones that had a longer neck ended up living longer. They survived long enough to reproduce, etc. Okay, so any change that confers an advantage during the selection pressure will be retained by the population, long necks and giraffes. So here's a little concept check. So which of the following mutations would cause a frame shift mutation? Okay, pause and come back. Well, remember, when we're shifting our frame, we're either inserting or deleting a letter. So this would be a deletion mutation. Wrapping it up, guys. There's only about 10, 15 more slides. Okay. So DNA recombination events. Genetic recombination occurs when an organism acquires and expresses genes that originated in another organism. And three means for genetic recombinations in a bacteria are going to be conjugation, transformation, and transduction. So with conjugation, the transfer of a plasmid or chromosomal fragment from a donor cell to a recipient cell via direct contact. So this would be like when a gram-negative cell donor has fertility plasmid or F factor that allows the synthesis of a conjunctive pilus. Then the donor, which is an F positive, transfer for fertility plasmid through a pilus to the recipient, which is an F minus cell. And then this therefore becomes an F plus cell. And since some F plus cells become HFR cells, which would be high frequency of recombination. Okay, so here is a fun little figure of physical conjugation. And this is figure 9.24. And so you can see here that the pilus of the donor cell on the top attaches to the receptor of the recipient cell, and it retracts to draw the two cells together. So it's like, hey, come here, right? And then this is the mechanism for gram-negative bacteria. And then again, they have high frequency of recombination, and this is because the donor's fertility plasmid is going to be integrated into the bacterial chromosome. And then when that conjugation occurs, a portion of the chromosome and a portion of the fertility plasmid are going to be transferred into the recipient. So this is the second part of figure 9.24, and this is that transfer of the F factor, or the conjugative plasmid. Okay, so you can see they attach together, and then they're going to like spit it out, and then they transfer some of that material. Okay. And then this again is step 3 of process 9.24, and this is the high frequency transfer, or HFR. And this involves transmission of chromosomal genes from a donor cell to a recipient cell. And the donor chromosome is duplicated and transmitted in part to the recipient cell, where it is integrated into the chromosome, or the genetic material of that cell. And then the chromosome fragments from a lysed or exploded cell are going to be accepted by the recipient cell, and the genetic code of the DNA fragment is acquired by the recipient. So the donor and recipient cells can be unrelated, and this is a very useful tool in recombination of DNA. So here is a fun picture of transformation, and this is shown in figure 2.26. Okay, and so you can see that up here, the DNA fragment, which is in blue, delivers the cap positive gene for the capsule formation, which is in red, and it binds to the surface receptor of competent recipient cell. And then the DNA is going to be converted to one strand and transported into the cell by the DNA transport system. And then the DNA strand down here aligns itself with a compatible region of the recipient chromosome. So you can see this area is aligning itself. And then the DNA strand here is going to be incorporated into the recipient chromosome. And now the recipient is going to be transformed with a gene synthesizing a capsule. Okay, so this would be the transformed cell. And so that's what I just told you. Okay. So then this would be looking at Griffith's work in transformation, and this is the start of figure 9.25. And so in essence, this experiment proved that DNA released from a killed cell can be acquired by a live cell, and the cell receiving this new DNA is genetically transformed, in this case from a non-virulent strain to a virulent one. So you can see the cell type, when it's transferred to a live mouse, it can cause it to die, where if it has no capsule, then that's going to be where it's going to survive. Okay, so again, that's figure 9.25. Same here. So this is the bottom of it. When you have a heat-killed S strain, you give it, it can survive, but when you have the live plus the heat-killed and you transform it, it can die because the live S and R strains were isolated from the dead mouse. So then you have transduction, okay? And the bacteriophage serves as a carrier of DNA from a donor cell to a recipient cell. And you have two different types, either generalized transduction, which is random fragments of disintegrating hosts of DNA are picked up by the phage during assembly, and any gene can be transmitted this way. And then you also have specialized transduction. This is a highly specific part of the host genome, which is regularly incorporated into the virus. Okay, and these are pictures of 9.27 and 9.28. Okay, so you can see this one's 9.27. So this is the generalized transduction, or the genetic transfer means of a viral carrier. So in step 1 up here, a phage infects the cell, A, which is the donor cell by normal means. And then 2, during replication, in assembly, the phage particles incorporate and segment of bacterial DNA by mistake. And then step 3, cell A then lyses and releases the mature phages, including the genetically altered one. Okay, so this is step 3 right here. And then step 4 is down here. Again, this is figure 9.27 in your book. The altered phage absorbs into and penetrates another host cell, B, injecting the DNA from cell A rather than the viral nucleic acid. And then down here in 5, cell B receives this donated DNA, which recombines on its own DNA. And because the virus is defective, meaning biologically inactive as a virus. It is unable to complete a lytic cycle, and that the transduced cell survives and being used as this new genetic material. Over here, this is figure 9.28. And this is specialized transduction, where there's a transfer of genetic material by the means of a viral carrier. So here in 1, you have a specialized transduction that's going to begin with a cell that contains the phage, which is going to be red. Okay, and that's over here in this little area. And then 2, when the virus enters the lytic cycle, it excises itself from its host cell, carrying along some of the host DNA. And then in 3, the replication and assembly results in the production of a chimeric virus, which contains the virus and bacterial DNA. And then down in 4, you have the release of the recombinant virus and infection of a new host that results in the transfer of bacterial cell DNA between cells. And then in 5, which is down here, the recombination can occur between the new host chromosomes and the viral DNA. This results in either bacterial DNA or a combination of viral and bacterial DNA being incorporated into the bacterial chromosome. Okay. Table 9.5 here are the types of genetic recombinations that occur in bacteria. So that's important to study. And then moving on and finishing up, we have transpositions. And this is special DNA segments that have the capability of moving from one location of the genome to another. So they like jump genes. And these cause rearrangements of the genetic material, and they can move from one chromosomal site to another. They can form a chromosome to a plasmid, or they can be from a plasmid to a chromosome. And this may be beneficial or harmful. It just really depends on the situation. And so this is figure 9.29, and this is a shifting segment of the genome. So up here in 1, you can see the transposition exists as a small piece of DNA is integrated into the host of the chromosome, and that's the red part. And then down here in 2, the transposition may excise itself and move from one location to another in the genome, maintaining itself as a single copy per cell. Or 3, it may also replicate prior to moving, leading to an increase in the copy number and a greater effect on the genome of the host. And then 4, finally, the transposition may jump to a plasmid, which can then be transferred to another bacterial cell. So, tidying up here, guys. The genetics of animal viruses. We have a viral genome, and this is where we have one or more pieces of DNA or RNA that contain only genes that are needed for production of new viruses. And those viruses require the access to a host cell's genetic and metabolic machinery to instruct the host cell to make new viral particles. Because remember, that's technically why viruses aren't living, because they can't reproduce on their own. They have to take over, hijack a cell. Most viruses contain normal double-stranded DNA or single-stranded RNA, but there are other patterns that exist which make viruses very unique. Okay, with a few exceptions, the replication of the viral DNA occurs in the nucleus, and the genome of most RNA viruses is replicated in the cytoplasm. And then in all viruses, viral mRNA is translated into viral proteins on the host cell's ribosome using the host tRNA. And so this one is going to be figure 9.30, and this is the general stages in the multiplication of double-stranded DNA viruses. And this is where up here in 1, the let's see, the DNA, okay, so the virus penetrates the host cell and it releases the DNA, which enters the nucleus, and then it's going to be transcribed by the host cells into the mRNA. And then here, the viral mRNA leaves the nucleus and is translated to the structural proteins, and these proteins are then transported into the nucleus. And then down here in 4, we have viral DNA is replicated repeatedly in the nucleus because it hijacked and took over. And then over here in 5, the viral DNA and proteins are assembled into mature viruses in the nucleus. And then down here in 6, because it is double-stranded, some viral DNA can insert itself into the DNA host, and this is called latency. And there are some exceptions to this pattern, and these mostly occur in the pox viruses. Okay. What about the replication of the ssRNA viruses? Well, this is going to be figure 9.31. And so in general, these viruses do not enter the nucleus, where 1 here we have a penetration of the viral RNA. And because it is positive in the messenger and the single-stranded, the RNA can be directly translated on a host cell ribosomes into various necessary viral proteins. And then down here, that was 2 right here, sorry. And then in 3, a negative genome is synthesized against a positive template to produce large numbers of positive genomes to finally assemble. And then down here in 4, the negative template is then used to synthesize a series of positive replicates. And then down here in 5, the RNA strands in the proteins assemble into a massive virus that is released when the cell lyses or explodes. Okay, so we get to do a concept check, and then I'm letting you guys go. So, which of the following terms describes the genetic transfer of bacterial DNA that occurs by direct cell-to-cell contact and is mediated by bacterial plasmids? Think about it, pause it, and come back. Well, that would be conjugation. Okay? And so that is the end of the presentation. Thanks for coming along for this chapter, chapter 9, and we'll see you guys in class. Thanks.